So thanks everyone for showing up. My name is Lisa, like Samantha said, and I'm a PhD candidate at McMaster in the Granfield Research Group. So I'm in my third year right now, and I'm also the current student representative of the Microscopical Society of Canada. So in my doctoral studies, I've been working with liquid phase microscopy methods in SEM and TEM. And today I'll be talking about some recent work that we've been kind of exploring in it as a team with room temperature ionic liquids and EM applications. And we'll focus more on SEM with some biological applications, but I made sure to include some TEM and non-biological applications of this technique as well in here. And just a little bit about our team here. So in my team, we focus on this kind of hierarchical theme of multi-scale microscopy of biomaterials. So we have three main focuses. We look at biomaterials, biointerfaces, and biomineralization. And we're located at McMaster University in Hamilton, Ontario. And so we're really fortunate because just in the basement of the building that we work in is the Canadian Centre for Electron Microscopy. So we have really fast access to the microscopes and all the amazing things and tools that they have there. So they've been really vital in all of my research. And of course, I was really happy to give this webinar today to help out. So in today's talk, I'll go through kind of a casual presentation talking about our work with ionic liquids. So I'll introduce this as a topic and maybe a little bit of why we're interested in using it and where it's been so helpful for us. So I'll go through some applications in biological fields, especially talking about SEM applications. And then I'll talk about some other applications and perspectives on the technique. So as many of us know, before we start, I think many of us know what this is. So here we have a scanning electron microscope where we have an electron beam. It's condensed through a series of lenses. It's rastered and interacting with our sample with this fine probe. And then we get these redirected signals and electrons that we have detectors and things that form our images. So this is all really nice, and we're able to look at things at really high resolution. And for this all to happen, of course, sample preparation is super important. So I used to be a metallurgist. I had no problems with my samples when I was dealing with metals. They were dry, they were conductive and vacuum stable. I would abuse these samples with an electron beam back when I was in my master's and undergrad. But now that I've been working with biological samples in their natural state, these are soft, hydrated materials that are acting as insulators, and they tend to be electron beam sensitive. So largely they are challenging to image in their natural state. If I were to just put a biological sample in the microscope, like I used to do with my metals, you'd have charging effects, degradation, vaporization, shrinkage, likely contamination in the system. And there are alternatives of how you can look at these samples in their natural state, like environmental SEM or a more representative state in cryo-EM. But if you're looking at conventional SEM, you do need to do quite a bit of prep work to get your biosample into a regular SEM. So here I provide a really exaggerated example, but this is how a day of the life in the Granfield Research Group often works, is we work with a lot of bone samples. So this involves us selecting our region of interest, cutting our sample. This is normal stuff when you're preparing your samples, but then there's many lengthy steps that we have to do to get it stable in an SEM. So for instance, if we're taking this small piece of bone, it may take up to 48 hours to fix it. So we use this glutaldehyde fixation process. It cross-links the chains and sacrifices the natural fluidity of our structure to kind of stabilize it and keep it steady. And then after these kind of processes, we would normally, so that the sample doesn't evaporate in the chamber, have a series of dehydration steps. Again, for a small piece of bone, this could be a very lengthy process. And if you're dealing with anything larger, these numbers could quadruple almost in length. And so after that, we would normally do a critical point drying step. And again, you're drying your sample. With bone, it does have a rigid structure, but when I'm often talking about this with my partner, we give this analogy. I'm always telling him about Jello. And I'm like, well, if you're looking at Jello, which is analogous to a lot of gelatinous kind of biomaterials, you're looking at Jello in its hydrated state versus its dehydrated state. The morphology is completely different. So that's something to keep in mind when you're dealing with these dehydrated samples as well. So again, we have it dehydrated now, but they're not conductive, so we have to do a sputter coating. And this is a pretty standard process in most EM prep methods. But for bone, especially if it's not embedded, it may be porous, and the in the sputter coating process is more or less a line of sight process. So you could have pores that are completely not sputter coated or not well coated, which can affect overall the quality of your imaging. So at the end of the day, after all these steps, we can finally look at our sample in SEM. And these can lead to, after all the lab bookings and the different running around to different things, this can lead to hundreds of hours of lab prep time. And this is for, again, a very small sample. If we were dealing with a whole femur fracture or something like that in a biomechanical study, we don't have an easy way of getting it into an SEM. So very challenging, long works. Every time we have students in our group preparing bone samples, they are complaining. But in any case, so this kind of brings us to these following questions. Knowing all this now, for those of you not in the biological field, when we're doing all these preparation methods, at the end of the day, after all these conventional methods, we have to ask ourselves, are we really looking at naturally occurring features? And how much are our preparation methods affecting our samples? And this is not necessarily just an application to bone. I gave that as an example that's really exaggerated with the times. But for most biological samples being observed in EM applications, as well as non-biological materials that are being observed in a dehydrated state. So these are questions in my research I'm trying to shed a little bit more light on when we're looking at these liquid or wet phase microscopy methods. And it's not the all solution artifact free method, but it does remove the kind of artifacts that may be coming from sample preparation and provide us a correlation to what we may have been seeing in an alternative state. So a quick plug-in to our research team. This is some of our current in-house liquid phase capabilities that my group works with. So we have access to a number of different tools to do low macro to nanoscale liquid phase imaging, which can enable us to observe wet hydrated biological samples in native hydrated environments and observe in situ reactions taking place dynamically. Now, while we can do some really cool things with these processes, they're often costly. You can only use them one at a time. They're not reusable, and they really do impose a lot of limitations on your sample size. So this is where we kind of led into this room temperature ionic liquid theme. So to address this long introduction, not everyone has access to these expensive equipment, or we don't want to use them all the time for every kind of study that we're doing, especially with these larger samples. So in this case, people are somewhat turning towards these room temperature ionic liquids as an alternative for hydrated imaging methods. So they are composed of ions and short-lived ionic pairs that are liquid at room temperature, and they're known for their low vapor pressure, high ionic conductivity, non-combustibility, and they're also really good solvents. They can dissolve a number of substances, and they're often referred to as designer substances or designer solvents. And their virtually negligible vapor pressure is due to strong ionic interactions, while their asymmetry contributes to their liquid nature. So typically you have an ionic liquid that has at least one ion with a delocalized charge and one component that's organic. So this prevents really the formation of a stable lattice. And since their conception, a variety of commercially available water-stable ionic liquids have been developed using a variety of catonic cationic and ionic and anionic combinations. So this gives them a unique subset of physiochemical properties you can achieve by combining different ions and cations. And they're used in a lot of different materials applications. So before I was using them in EM, they're often known as solvents, electrolytes. They're used in liquid crystal applications. Here, the director of the CCM, Nabil, he had past research and a patent using ionic liquids for 2D material prep. So when I first was talking about this with him, he had all these questions for me. He was so excited to hear me even mention the word ionic liquid because it was a bit of a blast from the past for him. So in any case, going to the electron microscopy things here, in this 2006 work by Kuwabata and al., they were the first to show that these ionic liquids were really stable in SEM. And I call these the brave souls that actually put wet things in their microscope and tested it out for us. And so they showed that at normal operating conditions, these ionic liquids were stable. They actually even rotated their stage to show that the liquid was moving a little bit. And then they also showed in this paper that these artils behave like conductive coatings and they experience limited charge buildup. So in this case, they have these star sand grains that they dipped one of them in and the other they didn't dip in an ionic liquid, and they're showing that it can be used like a conductive coating analogous to that. So in this case, this really provides us new avenues for potential electron microscopy work involving dehydrated biological materials as well as in situ observations, because we know this liquid's stable in vacuum, and if we can hydrate a material with it, then we can observe it in SEM. And as well, we found that in a lot of literature, it does provide similar or comparable, I should say, a resolution in contrast to conventional methods that are being used for biological imaging. So I have this wonderful paper that I show on this slide. This is from Golding and al. They're from Manitoba, actually. So other Canadian researchers, which is kind of cool. But here in this work, they're looking at microbes, and these are totally unfixed and hydrated microbes. I don't know how they got Ebola into a microscope unfixed, but I know they have those really high BSL labs out that way. But in any case, here in this work, they're showing different preparation techniques. So how like the typical dehydration sputter coat compares to an ionic liquid prep to a negative Stamen TEM. And what they know is they got comparable imaging. Obviously, looking at something wet, sometimes you're not going to get the best of the creme de la creme that you can get when something's sputter coated with a really nice conductive coating, and as well as in TEM too, you have quite the advantage there. But the key note that they show is that they can prepare their samples very quickly. In under an hour, they could get their samples in the microscope. And when they were doing some on one of these microbes, they had done some measurements of the morphology sizes, and they noted there was a better conservation of the natural size and morphology of their samples in comparison to past cryo-EM work that they had done. And again, there's a mitigation too of some sample defects that seem to come up from the preparation methods, such as like flattening of the samples or dehydration effects. So that really helped inspire me and get me interested in a lot of biological ionic liquid work. So we researched this technique more, and we found this is a typical application process when you're using ionic liquids in EM applications. So here I'm specifically showing for SEM, but I do have some TEM stuff coming down the line. And so in this case, you typically have an ionic solution that you're preparing. Normally, I see this in percentages from 1 to even 20% volume percent of an ionic liquid to an immersion solution like water or ethanol. And you can either add the sample directly to your sample that you're observing, or you can immerse it within there. Typically in our lab, when we're doing this addition sort of technique, we'll have a piece of like a biological material, we'll add our solution in, hydrated it for about 60 seconds, and then we remove excess solution with filter paper. We typically also heat the solutions before we apply them. So we'll add it in a water bath at about 40 degrees or even 60 or 70 degrees. And this helps improve the spreadability of the solution because it is quite viscous in nature. And then prior to putting it in the SEM, we do sometimes do like a vacuum treatment just in case there's any excess solution being left on the samples. We'll either put it in a desiccator or a vacuum environment for 10 to 30 minutes. And at that point, we would put it in the SEM. And this has been shown in literature to work for a variety of samples. So if we're talking about biological samples, they've done it with fixed or unfixed samples, stained or unstained samples. And for other non-biological samples, people have done this for dry samples, soft or wet samples, or liquid samples. And again, if we're talking about the research we just showed on the previous slide, you can get improved. When we're saying improved imaging quality, we're maybe referring to like the overall quality and mitigation of some artifacts from preparation, or similar imaging quality to traditional techniques. So in a first study that my team did, after hearing about this technique, we really wanted to try it in our lab. You saw how long it takes us to prepare bone samples. If we could shed any of that time for sample prep, it could really benefit our team's work, especially with biological or soft materials requiring dehydration. So it was a past PhD student of ours, Brian Lee, who led this work, and he was working a lot with these cells. And what he noticed is the normal preparation methods, when you're looking at cells in EM, they typically shrink cells, they alter their natural fluidity and morphology, but it's a little more than that, because I've helped prepare cells as well to be observed in SEM, and sometimes you might have these beautiful results showing like exactly what you want from your surface. Then you prepare your samples and you get to the microscope and all your cells have exploded for some reason on the surface, or they don't look quite right after the preparation. Or like maybe they've all disappeared. Sometimes they just lift off randomly. So and it's often like I'm still learning as well preparation methods, but sometimes it just feels random. Like during the preparation, your samples just disappear at some points, and this is normal. Unfortunately, sometimes things just don't work out when you're preparing your cells on a surface. So Brian had this idea, and he was working with cells on laser structured surfaces. So he tried this RTL method, and he used this really common RTL here with his SEOS2 cells, which are like bone-like cells on laser modified surfaces. And he developed this protocol, and instead of doing what literature was doing normally, he was mixing his ionic liquid with cell culture media. So he actually was able to keep his cells alive during the treatment, and they were alive like putting them into the microscope, and on removal, the ones that were irradiated by the electron beam were not alive, but there were some activity still on the samples, and it showed that the ionic liquid cell culture media wasn't cytotoxic. So that was really cool, because we could see these cells in what would be like the last natural kind of interaction that they've had. And so he noted in a lot of his preparation methods, when he was looking at these samples dehydrated and conventionally prepared, he always saw the cells were aligned in like a perpendicular way to these laser modified kind of surface topographies. But when he looked at them in the ionic liquid, they were aligned actually more in parallel with these modified surfaces. So it was a little interesting observation he had. And he showed that this was in backscatter mode here, and he's showing that you're able to get sufficient contrast and charge dissipation for SEM of the cells on these nanostructured surfaces. And again, these are unfixed, fully hydrated cells. So Brian did a lot of work with this, and when he left the team and graduated, I took over and I started looking at this protocol in a little bit more detail. And so we were able to adapt it and avoid all these laborious things. So we had this success with using ionic liquids. And so I started with the team. I got a little bit of training with Brian before he left, and then COVID-19 happened. So this led us into a deeper dive into the technique, and we jumped into a bit of a rabbit hole of literature at the start of COVID-19. And so I had this outcome, I call this my COVID baby, but this was a review paper we put together, because what we were finding is when we were trying to look at ionic liquids and how to apply them in EM applications, we weren't able to find this summary of best practices. And it was all very almost seemed arbitrary, the way that people chose their parameters, and we weren't sure what worked best. So in our group now, we do perform a lot of protocol development and we work on this, but this review paper helped us summarize some best practices and applications on where to start for us, because we had no idea otherwise, other than the protocol we were using, but we wanted to know about more ionic liquids, more methods. So our review has, for anyone looking to use ionic liquids, it's a really good starting point for researchers in this field. I summarize a table of commonly used ionic liquids in imaging applications, and then I go through another table with treatment parameters for various categories of biological materials found in literature. And this has been really useful in our team, not just for biological imaging, but we often look at this table and we're like, okay, this is this material, it reminds me of this material, so this treatment should work out well, or is a good starting point for us. Some snippets of biological applications, imaging applications that people have done with ionic liquids, I have on this screen. So here we have this Suda and al, and they're a big group that's publishing in this ionic liquid imaging field. They're often developing ionic liquids. They're more on the chemistry side, but here they have a yellow jacket that they imaged in SEM with an ionic liquid treatment. In this figure, they're showing various pollen grains. So a key part here is the morphologies of these samples. They have really sharp features, and especially with these really star-shaped structures, they found that it's really hard usually to mitigate charging and those bright edge effects at really sharp features. And it seemed that in their research that using ionic liquids helped mitigate those edge effects. They're not completely gone, but in comparison to conventional methods, it did help a little bit with these sharp features. You have works as well. There's a lot of people imaging wood with ionic liquids, and that I found very interesting because it has an analogous kind of hierarchical structures to bone in my own research, as well as cells. Cells are a very common application, and it really does make sense for especially people in the biomaterials field that are looking at cells to study these cellular interactions in the hydrated state, because just like Brian was dealing with, if you're doing these conventional preparations, some papers kind of hinge on what they're observing in EEM, which may not be representative of what's actually happening in the hydrated state. So these are all some kind of snippets of biological applications I found in literature to show. We have a little bit of work that I'll just give you a snippet of, which we're publishing now with ionic liquid treatments. So going back to bone, we did use this on bone samples. So I developed a protocol off of the protocols we've seen in other biological materials. This was a first time application of this that we found in literature. So starting again, you cut your region of interest, you segment that bone sample out, and then you can go and skip the dehydration step, skip the fixation step, and directly apply your ionic liquid solution to this. So in this case, we used a water solution and we varied it from 1 to 20% just about. And all we did is we exposed the sample to the solution for 60 seconds. We blotted the sample to remove excess solution afterwards, and we vacuum treated the samples in a desiccator before putting it in the electron microscope. And this could be done within 3 hours, which is really amazing because we could get pretty much a bone sample in the morning fresh and then image it that afternoon. So this enables us to do this. We looked at this study, we used 2 common ionic liquid treatments. So we had this BAMI-BF4 and this ME-BF4. These are 2 really common imaging ionic liquids that are used in application. And because no one had done this before, I had to kind of play around a little bit more with the parameters, but it was more of a feasibility study to develop the protocol that would work in our team to try different things. So in this case, I just have a snippet of some of the images I have from this treatment in backscatter imaging, and it worked. So I was really happy with this. This is on our Joule 6610 microscope here. We found these are healthy bone structures shown. So you have cortical bone and trabecular bone. Cortical is this compact bone that's like more dense, whereas trabecular bone has more free floating porous structures. And so we could make out really key features that we're always looking to observe at in SEM. So you have these osteons, which are surrounded by these blood networks. It's these laminar kind of bone structures that surround these blood networks, as well as these osteocyte lacunae networks. We could be able to spot them in the SEM. We did find an optimal treatment. This is the treatment that behaved the best. It didn't have pooling or liquid pooling at the surface, for instance. It imaged quite well, very consistently. We were able to use this in both low and high vacuum imaging conditions with at a kind of normal voltage of 15 kV. And when we were doing this, this was one of the first studies we did in high vacuum. So we wanted to take a lot of baby steps and make sure that everything was going to be safe along the way. So we did low vacuum, and then we did our observations, and we switched to high vacuum. We found that it could work in backscatter and secondary electron modes. So the secondary electron mode was really useful for looking at fracture surfaces. We had a piece of bone from a collaborator of ours that they used in a biomechanical study we were able to look at with this treatment. And we found that there's a mitigation of sample preparation defects. And what we normally found in the conventionally prepared samples is you had a lot more micro cracks, like little cracks appearing from the dehydration, where these are almost not even present in the ionic liquid treatment samples. We mostly just see naturally occurring cracks in these surfaces. So that was a really useful observation. And as well, we found with the trabecular structures, these are unembedded samples. So these free floating structures don't really have a good conductive line to the bottom of the SEM stage. So in conventionally prepared methods, these trabecular structures were often even difficult to look at. But with the ionic liquid treatment, we could make out really nice features and had no issues with charging. So that was a really cool study we did. We've also used these to look at biofilms. So biofilms are really, really impacted by dehydration and harsh fixation methods. So they're very gelatinous. So we can go back to our Jello methodology or our Jello analogy, and it's really like you're looking at two different structures when it's hydrated versus dehydrated. So in the conventionally prepared sort of biofilms you see in nature, when you're looking at normal papers, they often have this fibrous biofilm, and it's kind of looks a little exploded almost, whereas Asahi and all in this work, they're looking at strep, and they did do an ionic liquid treatment here of this really thick, kind of gross to see such a thick biofilm, but this really thick biofilm. And in the hydrated versus dehydrated states, it's really quite a different structure. They're much more compact in the RTL treated structure versus the conventionally prepared structure. So we tried this ourselves, and we're developing a protocol that we're working on publishing right now. But this is just a snippet. And in comparison, we're looking at staph and Pseudomonas. And these are two really common clinically relevant biofilms. And here in the dehydrated, conventionally prepared gold-coated structures, you see fibrous structures that are kind of disconnected. There's little things here and there about them in holes. Whereas in the RTL treated structures, especially with the Pseudomonas, the structure is completely different. It's very, very compact. So it was really, it was really interesting to see that difference, as well as with the staph too. It almost looked like they're buttons versus spheres, and we were comparing them all to food, for instance. So with the Pseudomonas, it looked like Jolly Ranchers versus here it looks more like a gnocchi. But yeah, it's really interesting to see, and we've been working on this, and the comparison to conventional methods has been really drastically different. And as well, here the images we're showing are in our field emission SEM at really high vacuum condition. So we were able to push that, and it was really stable in that condition. So that was really an important observation for us to make in our future studies. We've been using a lot more FEG SEM things in these kind of studies that we've been doing. So I promised I'd show some more applications. So here I have a TEM example that someone's done. So this is Suda and al. I mentioned that they published a lot in this field. So this is showing a methodology of how you can use ionic liquids in TEM. Now I have not quite had the chance to try this at TEM yet. I haven't been brave enough, to be quite honest. But in any case, they show a really simple methodology. They have the membrane that they have, they deposit a liquid solution and let it rest. And this liquid solution contains their colloidal specimens and a little bit of ionic liquid. Then the samples sort of get in the holes of this colloidal membrane, and then they remove excess solution and dry, and they put it directly in their TEM. So the idea here is these holes in their TEM grid end up collecting the liquid with the specimens. So here they were able to look at these herpes simplex virus type 1 samples. There are quite different morphologies, and they talked about this in the paper where some of them may have been artifacts from the solution themselves, but in B, A, B, and E, you could have quite you have structures you can identify features from that look similar to the structures that they're expecting. Some non-biological applications now for those of you not in this field. I kind of summarized a few of them that I found interesting on this slide. So one of them I have is these SEM of hydrous super absorbent polymer beads. So this is really interesting to see, one, that you can use this for observing polymers, and they have pretty good contrast and resolution that they were able to achieve. These are from different ionic liquid treatments that they did, but D and G were quite similar to like what they were conventionally seen, but the size was more well conserved. Whereas A was really interesting because they were actually having an interaction with the polymer to the ionic liquid that they're using. And so that's just gives a little bit of insight that when you're using these kind of techniques, you have to consider how your solution might be actually interfering with your sample itself. This was a really neat one that I just stumbled upon the other day, actually. And this was from the Microscopy and Microanalysis Conference. So I only have the abstract and a few of their images that they showed. But here they're using this Hitachi Hylem ionic liquid. And in this case, this is a trademark ionic liquid that Hitachi makes that they won't tell anybody what's in it. But in any case, they were able to do tomography of gold nanoparticles in solution of these. Again, I didn't see any reconstructions from this work. Maybe they gave it in their presentation, but this was really interesting to see that it was stable enough and things weren't moving quite as fast that they could actually capture tomography with this. A common non-biological application of ionic liquids in electron microscopy applications involve electrodeposition. So here they're looking at these time points where they have silver nanoparticle clusters kind of growing in a custom electrochemistry setup. They had these grooves that allowed them to put their a charge through the system, but it was open to the environment, so they had their ionic liquid flowing through. And so that was really interesting to see, and especially because a lot of ionic liquids are used in electrochemistry applications, so it makes sense to be able to use them as a medium to observe them, observe some sort of reaction happening in situ in SEM. One last paper I found, which was really cool, this is from actually fellow Canadians. So this is Reynold Gauvain's work from McGill. So he's a frequent speaker at the CCM. So when I found this paper of his, I'm like, oh, that's so neat. Reynolds did this too. But he's got this really nice paper where they're looking at this rare earth ore, and they have wonderful SEM images in there. But what was really neat is they were able to get some EBSD mappings of there. So they have really nice band contrast and inverse pole figure maps, as well as phase maps that they were able to achieve with the ionic liquid treatment on these non-conductive kind of earth mineral structures. So that was a really neat application I've seen. So to date, I've been talking about ionic liquids and all these great things that it can do. And it probably sounds like I'm selling snake oil at this point, but there are some shortcomings to using our ionic liquids. And this is something that everybody should be made aware of if you're trying to explore this. The first key thing is it's not a preparation method. So when you're using this as a method for treatment, you can normally get like only a few days of life from these samples before maybe they're starting to degrade because you're keeping them in this wet state. So we in desiccators, we've been able to keep structures pretty nicely for up to 7 days. So cell structures and biofilm structures, maybe the bone would last a little bit longer. And then I know in literature, people have shown up to a month in a nice desiccator kind of safe environment. But again, it's not a permanent preparation method. So you can't go back to this a year later when a reviewer asks you to do something else. You'd have to redo your whole experiment. So that's something to keep in mind. There is quite a bit of technique apprehension to this. Mainly people are afraid of putting wet things in their electron microscopes, and that's with good reason. If you don't do this in a really slow and cautious way, or you don't follow what others have done in literature, you could be potentially introducing excess solution to your microscope and leading to some damage later down the line to your system. So again, going really slow and steady with this is really important. We always do a safety kind of vacuum check of our samples beforehand, where we just leave it in vacuum so we know excess solution that would evaporate in system would evaporate out of system. When we're doing it on a microscope with low and high vacuum possibilities, we always do low first, and we check the surface, make sure there's not excess liquid that's pooled onto the surface that would cause us trouble in high vacuum. And when I'm on our Magellan, we have a quick loader system. So when I'm doing studies there, I'm often either looking at the sample before in another microscope before putting it in, or I'll put the sample into our quick loader, leave it under vacuum in there for a little while, and then load it into the chamber. So there's a few things you can kind of take to slowly get used to this technique and use it more commonly, but it's something to keep in mind. And being afraid of putting wet things in a microscope is completely normal when you're dealing with these equipments. And there is this potential excess of liquid pooling on the surface. So what can sometimes happen if the sample solution doesn't spread evenly on the surface, it is quite viscous. So you can get pooling on structures. We recently had a hydrophobic surface that we were looking at that someone didn't tell us it was hydrophobic. And so you had these little droplets everywhere. And you can mitigate that with the right selection of an ionic liquid too, choosing if you have an hydrophobic surface, you can use an ethanol RTL or an RTL that's actually hydrophobic as well to match better with that surface characterization. But in any case, what these pooling can lead to is poorer resolution because the thickness of that film is so thick, and imaging artifacts. And the last thing is there's limited available literature that really shows all these applications and optimization methods that you can use with little to no repetition studies. So a lot of the papers you see in this field, they're still looking at the technique itself on new materials and not in regular kind of lab research where in the methods, it's just like a quick blip that we used ionic liquid treatment to image our samples. A lot of the literature is still looking at how to optimize this. And even with our review paper that we put together, there's still not a really good resource of showing how to choose ionic liquids optimally for kind of a wide variety of applications. So a quick outlook on what this looks like, what the future of ionic liquids look like. There's still a lot of applications that have yet to be explored using these methods, and there's room for research. So even in RT, when we started this work, nobody had ever looked at using this to image bone. So that was something that we kind of explored ourselves, and there's very specific applications. I know, for instance, like there was a study where there's like a water sensitive metal, for instance, and the CCM was looking at exploring ionic liquids for that. But there's other applications too, and not a whole lot of people have done this kind of research. There's a need for these overarching principles and best practices to be summarized and developed to understand this kind of methodology a little bit better. And this will help standardize this process. And at the end of the day, we need to look at more of these treatments, and this will aid in its wider adoption and as a technique itself. So as a quick summary, I hope I was able in this webinar to show that our TILs offer an efficient and novel time-saving way to observe natural hydrated biological samples in electron microscopy, and not just biological samples. There's a wide range of other applications that this can be used for, especially when we're talking about looking at in situ reactions or dehydrated samples and looking at them in their natural state. A key thing that this method can do is mitigate sample damage arising from preparation and save you time on these laborious kind of conventional means of preparing samples. And we found that you can get kind of comparable imaging with the right protocol optimization in using this methodology. So thank you all for attending my talk. Just a few acknowledgements. I have to thank the CCM for inviting us here and for their support in all of our research. They've been really nicely supporting these ionic liquid studies we've been doing, and McMaster University and NSERC for funding. Of course, my research group for their support, and my advisor, Dr. Catherine Granfield, has been really wonderful, and all of our collaborators that we've been working on with ionic liquid research. And before I end, I just wanted to chime in that as the Microscopical Society student representative, we're hosting this event coming forward. So this is an event November 5th called Finding Your Path in Microscopy. So this is a virtual career event that's aimed for students looking to explore kind of some life advice and career advice. So we have a really exciting lineup of panelists, including Samantha Stambula from the CCM. So be sure to register and come out. It's going to be a really nice event, and we're going to be announcing some new exciting initiatives that are student-centered from the Microscopical Society of Canada, including new student chapters. So it's going to be really exciting. I'll put the URL in the chat before I head out, and that's it. So thank you everyone for your attention. I really appreciate you coming out here today.
Lisa. And again, if anyone has any questions, feel free to put them in the Q&A or the chat. So one question, Lisa, what would the cost of the ionic liquids be?
So yeah, one thing I didn't mention is they actually are quite inexpensive relative to like what you might invest in for all these other treatment processes. It does vary ionic liquid to ionic liquid, but some of what I've been using in the past little bit, maybe it was like what you would compare to normal chemical costs, like $50 to $200 for like a vial that we can use in large quantities. And often when I'm preparing an SEM stub, maybe I'm using like 50 microliters of a solution with like prepared with 10% ionic liquid. So I'm really conserving this ionic liquid, and we're not using it in large quantities.
Great. And in terms of, say, somebody that wants to try this for the first time, I know you've mentioned your review paper, but do you have any other suggestions, maybe just from your own experience?
So starting with like the review paper is it has those tables that can help indicate you where you can go, but trying to find something that's similar, analogous to your structure would be really important. So when I was looking at bone, we didn't have anything that we'd seen in literature of like mineralized tissues or things. But for instance, I found these wood papers that had these interligulous hydrated structures, and they had quite a few trial and efforts that they'd done for microscopy applications. So that was something that I ended up referring to quite a bit. And there are some, I mentioned quite a few times that Suda author and all in my work, and they have some wonderful papers, especially Kuwabata and all that did that 2006 work. They have some nice papers where they're exploring ionic liquids.
You mentioned during your presentation that even when in the SEM and in the ionic liquid, you could see that some of the samples were damaged after imaging with the electron beam, which is, I mean, expected, especially for biological materials. Did you find it was any slower or like lower rate of degradation or anything like that compared to like the dehydrated samples?
So this is something That's really depends on how the beams are radiating these samples. So in the hydrated state, you're getting a lot different interactions than when it's dehydrated and gold coated. They tend to be more stable after fixation because they pretty much go from a soft state to a hardened state, and then they're coated. Whereas in the electron beam, you definitely, in the liquid state, you're definitely getting different interactions. One thing that we did, that Brian did when he was doing that research, was he ran assays post situ. So he had these biological assays to kind of determine what the activity was and whether the cells were alive or dead or not. So that's what we ended up using to determine that. But you could, like, when I was looking at a surface recently that was hydrophobic that I talked about, when I looked at an area that was wet in SEM, you could see like bubbling, and I was like, oh no, that's not good. You can usually tell if something's not going right when you're imaging, so it's something to keep in mind.
Great. So what surface have you been growing your biofilms on, and can these solutions be used for colony biofilms growth on agar?
So we've done this. We've been mostly working with these titanium and steel disc plates because we've just been optimizing the protocol. But the idea would be that we can use this for multi-scale different biological materials that we're trying to do post situ, and look at cells, or post observation or experiment, and look at cells that are sitting on there naturally. We haven't done anything with agar. We did try on polymer surfaces before, and that worked okay. But we had other dramas there with conductivity in general with the polymer surfaces. But it's something to try. But one thing to keep in mind is you want to make sure whatever your substance is, your ionic liquid and solvent that you're using, it's not going to dissolve any of the materials you're using. So that's something to keep in mind. Maybe we've done before a test where we made our solutions and imaged it on the surface as a control, and then that way we had an idea whether anything weird was happening with the solution to the surface, and dried it after to make sure it looked fine.